Hi students, after my first video on capacitation, I read and went through your feedbacks. I went through your comments as well. There are few things you people have wanted to address in this lecture. So before going into the details of the lecture, let me go back to the previous class and then we will start this class. In the previous class, we started discussing about the word capacitation. Better I should say, this is sperm capacitation. In the capacitation, we introduced two words, that the sperm cells undergo physiological and biochemical changes. These physiological changes and biochemical changes make the sperm capable of fertilization. This we have discussed in our previous class. That means there are two phases of capacitation. I can use the word before capacitation and after capacitation. Before capacitation, the sperm is mature structurally and functionally, but it cannot bring out fertilization. So here a very simple question comes to my mind. If the sperm is mature, that means fully formed structurally and also functionally undergone a lot of modification in the epididymis, then why it is not capable of fertilization? Then the second aspect is after the sperm undergoes physiological changes, second biochemical changes, we will discuss today what are those changes, but after that it is capable of fertilization. So again the same concept comes to my mind, that what actually happens during these physiological and biochemical changes that this sperm is capable of fertilization. Let me draw another very simple flowchart to explain you what actually happens, what we need during the process of capacitation so that this spermatozoa is capable of fertilization. So I can write the word capacitation in the middle because it is that event where I can focus on two aspects. The first one is before. The sperm membrane, the first instance, the sperm membrane, the cholesterol, which is the part of the sperm membrane, after the process of capacitation, that is after, or you can say during, I can use the word during, there is removal of cholesterol. This is the first point I can say, that is removal of cholesterol. So what is the point of interest? A question mark here. How the cholesterol is removed? Why the cholesterol is removed? And what is the outcome of removal of this cholesterol? Second point. Here we can say there are some molecules which mediate removal of cholesterol. That we will discuss, and who are those molecules which are responsible for removal of cholesterol. The second point, influx of bicarbonate is almost negligible. Almost negligible. But during capacitation, there is influx of bicarbonate ions. Now the point is, why here? And second point, how here? One is why here, second is how here. And third, what is the outcome? What is the outcome? So this is our second point. The third point I can say, this is another very important aspect. Activation of soluble adenyl cyclase. Activation of soluble adenyl cyclase. Before capacitation, this enzyme adenyl cyclase is inactive. That is the actual point. That before capacitation, this enzyme soluble adenyl cyclase is inactive. It means now after or during there is activation of soluble adenyl cyclase. Now again three questions. How it will activated and what will happen if it is activated. So these are the three key points which are activated or which occur during the transition between before to after or before to during after. We will discuss one by one and will make another comprehensive flowchart to show how these processes occur. So in the next video, I will show you some other points, other 3 points, and then we'll combine the 6 points and we'll show how the capacitation occurs. Thank you very much. Go through this video and let me know if you cannot understand anything, and please try to comment in the chat box so that I can read and I can get back to you. Thank you so much for your patient hearing.